Okay, everyone. It's just a minute past noon. Actually, I need to put that presentation back up for 1 second. Kara McCreary, the greenhouse vegetable IPM specialist, is going to talk a little bit about the GrowON webinar series that you're joining us for. Kara, you want to unmute yourself first? There you go.
Thanks, Sarah. Hi, everyone. So I'd like to welcome you to the GrowON webinar series. And for those of you who are new to this, the GrowON webinar series is run by the greenhouse team at OMAFRA. So you can see the 4 of us on that slide. The 2 IPM specialists are the one hosting for today. And I'll let Sarah take over momentarily. But just to give you an idea of what we're trying to do here, our goal is to pool resources across floriculture and vegetable production to bring the greenhouse industry exciting talks on advances in technology, new production techniques, or novel pest management. So make sure to follow at least one of us, better yet, more than one of us, on LinkedIn or the On Floriculture blog or on Greenhouse Vegetables. And that way you'll get some updates on our upcoming webinars. So generally, these webinars are designed for all kinds of growers, from greenhouse veg to floriculture to novel crops like cannabis and vertical farming, and basically anything grown in a controlled environment. So with that in mind, I'll ask Sarah to launch the first poll so that we can find out a little bit more about who you are.
Yeah, so this should show up on your screen. If you just want to click, oh, I have to hit launch. It's a multiple choice answer. So if you just want to click the answer that best represents yourself, that will give us good information about the people that are joining us today, AKA who cares about onion thrips, which we really do. So you guys can see sort of the live poll answer. So I always love to see how it changes. I was like, ooh, consultants. Mm-hmm. Now it's like, now it's a little bit, more balanced. So it seems like we have, yeah, keep answering, everyone. It's great. Still going, still going. Are you guys changing your answers? I don't know. All right, so it seems like the number 1 is industry consultants. Next, we've got ornamental growers, researchers, and the cannabis and veggie growers next. So I'll close that poll out. I just wanted to quickly write some numbers beside that. Excellent, great. Oh, so you can actually share the results. So if you guys are just interested in looking at that for a second, this is the split breakdown that we're looking at in terms of who's joining us today. Okay. So I have the honor of introducing Ashley Summerfield. So she originally started off studying zoology, boo, and then was, luckily we got to have her. So she's been working as the senior lab technician for the biocontrol lab of Rose Boughton House at the Vineland Research Innovation Center for about 8 years. And then a couple of years ago, She came to me when I had a project going on onion thrips control, and she said, I really want to do this master's degree. So she graduated in April from her master's degree working on IPM of onion thrips. And also, I think one of the most important things she brings to our region and to our expertise in terms of IPM of ornamentals and other pests is, or other crops, is her amazing ability to be able to have the patience and skill to ID thrips. So we've been working sort of together on that, but there's a lot of me being like, okay, I don't know what this one is. Can you do this one? So she's a great resource to have in the region, and she's been helping me with lots of different projects. But today we get to hear about her master's stuff. So Ashley, if you want to start sharing your slides.
Okay, and I'll just cover quickly a couple of housekeeping notes. So just as a reminder, I'm sure most of you are familiar with this by now, but at the bottom of your screen, there should be a Q&A section. So if you can type your questions in there at any point through the presentation, we'll address them at the end. And the chat function is best to be used for more technical difficulties or general comments, and that will just help us coordinate the question and answer period at the end, if you could stick to those. So we'll hand it over to Ashley unless Sarah has any more comments.
No, I think we're good. Ashley, if you want to come on and just make sure you're unmuted, and I think we're good to go.
Okay, great. Yes, thank you both for having me as part of this webinar series. I think that it's a pretty exciting endeavor. So as Sarah said, I'm going to be telling you all about my master's research on onion thrips. And this is sort of an outline of what we're going to be talking about today. My thesis was really wide ranging because there's a lot of pieces that go into an IPM program. So I'm going to be discussing what thrip species we found in the greenhouse, infestation sources and timing. I did some testing of various biocontrol agents, so I'll tell you about those results. And I also did a little bit on pesticide efficacy and some compatibility with biocontrol. And then we will recap all of that, and hopefully all of this will come in a reasonable length of time. We'll see. Okay. Okay, so the majority of floriculture growers in Ontario have been using biocontrol as their primary means of thrips control for many years now. For the most part, the standard IPM strategy of predatory mites and Botanigard sprays have been very effective, but some growers have started to notice in the last 5 to 10 years that their IPM programs that used to work well for thrips just wasn't able to keep up anymore. In a recent survey that we conducted at the Vineland Research and Innovation Center, 57% of growers said that they noticed more frequent or severe thrips outbreaks in the last 5 years. So what changed? To figure this out, Sarah decided to have a closer look at the thrips themselves. Starting in 2016, OMAFRA began collecting and identifying thrips from floriculture greenhouses to verify exactly which species we had. And then I continued these surveys in 2019 as a part of my master's project. What we found is that although Western flower thrips still made up the majority of the thrip samples, on average, onion thrips made up about 30%. And this was a lot more than anyone expected because until this point, everyone just assumed that any yellow colored thrips in the greenhouse was Western flower thrips. Now, these relative proportions, so like this is an average, but they varied really dramatically depending on the location, which crop, and the different years. And in some circumstances, onion thrips can be the dominant species. And we don't actually know the reasons what makes onion thrips a major problem in some greenhouses or not. Although Western flower thrips and onion thrips look very much alike and share many of the same host plants, they do have a few key differences, particularly when it comes to their management. Biocontrol has been working pretty well for Western flower thrips most of the time, and that's good news because they are still as pesticide resistant as ever. Biocontrol hasn't been working as well, however, for onion thrips. They do appear to be more susceptible to pesticides compared to Western flower thrips, but they do also have a history of pesticide resistance, so there's no telling how long this is going to last. Greenhouses with high proportions of onion thrips also often experience more severe damage, but we don't actually know if all of the recent increases in the thrips outbreaks that growers have been experiencing are attributable to onion thrips or not, but I'm sure that is a contributing factor. So where did onion thrips come from all of a sudden? The truth is that onion thrips aren't that new at all. They've actually been established in Canada for well over 100 years. They used to be important greenhouse pests before the widespread use of pesticides in greenhouses. Since then, they've still, stuck around as a pest, primarily in outdoor crops, unsurprisingly in onions. And then Western flower thrips didn't actually start to take over the world and come to Canada until like the 80s and 90s. Thanks to their exceptional level of pesticide resistance, they became a major driving force behind the adoption of biocontrol in Canada. So I like to think that they helped in a very small way. In the early 2000s, though, changes in the pest management practices of onion crops led to increases in onion thrips pressure outdoors. By the 2010s, the majority of greenhouse growers were using biocontrol, especially for thrips. This increase in biocontrol use also came with a corresponding decrease in pesticide use. And it's my theory that the combination of increasing onion thrips outside, as well as a decrease in pesticide use inside, is what has led to their big comeback. But this is just my theory, and we don't actually know precisely when thrips populations in the greenhouse started to change, and that is for one very important reason. The reason being that no one was looking. The last time that greenhouses in Ontario were surveyed for their thrips species was in the 1980s, which means that there's a 30-year gap on our knowledge between the last survey and ours, so we don't really know what happened in that interval. One of the reasons why no one was looking is because with the naked eye, or even with a hand lens, all yellow-colored thrips pretty much look the same. Even a thrips expert can't confidently tell yellow thrips apart without a microscope. And it's because of this barrier that until very recently, most growers and consultants just assumed that yellow-colored thrips in the greenhouse were Western flower thrips. To identify thrips, you need to have a microscope and a taxonomic key. And this is another major barrier to identification. Most taxonomic keys are very complicated to use. They cover dozens or hundreds of species. The wording is really difficult to understand, even for me, and they use features that can only be seen under a very expensive compound microscope. But luckily, what we found is that the species that we typically find in greenhouses, we can tell them apart pretty reliably using a few simple features. So Sarah and I developed a key to species found in Ontario greenhouses. We designed it specifically for growers and consultants, and it uses features that you can see using a dissection microscope like the one in this picture. The most important features that we use in our key are the ocelli, which are these spots between the compound eyes, and the pronotum, which is the body segment below the head. Under a microscope, the differences between onion thrips and Western flower thrips are actually pretty simple. Western flower thrips have red ocelli and long hairs on both the top and the bottom of the pronotum. Basically, they're a hairy beast. If it's not a hairy beast, it's probably not a Western flower thrips. Onion thrips, on the other hand, have gray ocelli, and they're pretty much, most of the time, you just don't really see anything. And they only have hairs on the bottom of the pronotum, and those hairs are much daintier compared to the Western flower thrips. So like once you've seen this comparison side by side, it does get a lot simpler. And if you want to start identifying your own thrips, you can find our key on the On Floriculture blog. In addition to Western flower thrips and onion thrips, we also found chrysanthemum thrips, tobacco thrips, echinothrips. We found these pretty regularly in ornamental greenhouses throughout the region, although These species, they're usually show up in really low numbers, but I have seen each of them cause outbreaks, so it's worth being aware of their existence, particularly if you're seeing some unusual thrips activity. So for example, chrysanthemum thrips really hammers the bottom leaves of a plant. So it's really distinctly different from what you'd see with Western flower thrips. You can read more about the new thrips on the block in the February 2020 edition of Greenhouse Canada magazine. We've also found a few less common species, some of which are invasive. Most notably, Thrips parvispinus has recently popped up in a few greenhouses in the Niagara region and Essex County. By being aware of other Thrips species and having them identified, you're more likely to be able to spot these unusual Thrips like this yourself. And this allows you to intervene early and prevent them from establishing in the greenhouse. You can read more about the Thrips parvispinus in the September 2022 edition of Greenhouse Canada, and Sarah is also going to be giving a talk about them at this year's CGC. Besides intercepting possible invasive pests, why should you have your thrips identified? When we start to assume that Western flower thrips was the only thrip species, we forgot the first tenet of IPM, which is to start by identifying your pest. And as we've seen before with greenhouse versus Bemisia whitefly, or green peach versus foxglove aphid, you can't always use the same IPM program for all species of a given pest. And this seems to be the case for thrips as well. Misidentifying your pest means that you could be releasing bios that aren't having any effect, which is the equivalent of throwing your money away. So if your Thrips ID, if your Thrips IPM program isn't working as well as it used to, or you're seeing some unusual damage patterns, take a step back and make sure that you're dealing with the pest that you think you are. Now, growers don't necessarily need to ID the thrips themselves. A number of consultants have started to use our key to ID thrips for their clients, or you can send samples to Amafra and then we can help you out. Okay, so for the rest of the talk, I'm going to be focusing on the most common thrip species, which is Western flower thrips and onion thrips. And I'm going to start by talking about when and where they are coming from. Like any pest, thrips can enter the greenhouse in one of two ways. They can either sneak in on imported plants, or they come in from outside through openings like vents and doors. Several years ago, we did a study at Vineland to determine how many thrips were coming in on imported cuttings. We found that on average, there are about 5 to 10 thrips per 100 cuttings of chrysanthemums and spring annuals. And those numbers can really add up when you multiply them by thousands of cuttings every week. And that's ultimately adds up to a big problem. But unfortunately, we weren't paying attention. We were guilty of the same thing about making assumptions. We weren't super paying attention to which thrip species in this first trial. So we repeated it in 2019 with mum cuttings, and we did confirm that only Western flower thrips were found on these cuttings. So this suggests that cuttings are an unlikely infestation source for onion thrips, at least in chrysanthemums. That same year, we surveyed thrip species in chrysanthemum greenhouses in the Niagara region. We put up sticky cards both inside and outside the greenhouses. And thankfully, the identification features that we need to differentiate onion thrips and Western flower thrips are still visible on sticky cards. So we were able to separate out our card count data by species pretty easily. The majority of the thrips on cards were Western flower thrips, both inside and outside the greenhouse. In fact, there were actually twice as many Western flower thrips on the outside cards compared to the inside cards. And the number of the thrips that we collected on these cards outside indicates that there is a large Western flower thrips population in the landscape in Niagara. Now, this was a significant finding because when Western flower thrips first arrived in the region, researchers at Agriculture Canada determined that at that time, Western flower thrips wasn't able to overwinter in Southern Ontario. Evidently, they have adapted to our climate and they're no longer restricted to the greenhouse, at least here in Niagara. So in this graph, I've standardized the card captures by converting the counts to the proportion of total thrips that were caught per sample, per sampling interval. And by standardizing them, you can really see very clearly how the pattern, the seasonal pattern of Western flower thrips inside the greenhouse closely follows that of the outside population. And this tells us that the Western flower thrips in these greenhouses are mostly coming from outside. Before now, we've paid a lot of attention to stopping thrips, Western flower thrips that are coming in on plant material. And we kind of ignored the thrips that were coming in from outside. And many growers have adopted cutting dips, which is great. And that prevents, any of the thrips that you're importing from, propagators down south, possibly super pesticide resistant Western flower thrips. But clearly, based on this information, that's only solving half of the problem. Just like Western flower thrips, we also found onion thrips on both the outside and inside sticky cards. During the summer months, the changes in the thrips populations inside the greenhouse do closely match what was happening outside the greenhouse, albeit a little less perfectly than Western flower thrips did. But a notable difference between onion thrips and Western flower thrips, however, is that onion thrips continue to persist inside the greenhouse long after the pressure from outside died off. So based on this data, we can conclude that the outdoor thrips populations are most likely the initial infestation source for onion thrips. However, once they get inside, they establish themselves in the greenhouse and they stick around longer. In 2019, both thrips species peaked in July and August. So if you want to prevent thrips establishment, this is the key time to focus your thrips management efforts. And the best thing you can do to manage thrips, just like any pest, is to keep them out of your crop in the first place. Mass trapping is sometimes overlooked as an IPM tool, but it can prevent a lot of thrips from reaching your crop. Previous work by Rose Boutenhouse found that mass trapping is most effective at catching dispersing thrips, which means that they're actually really well suited to intercepting these thrips incoming from outside. So my advice is to go bananas with your mass trapping in the summer, especially in July and August. Research by Omafra found that wet glue type cards continue to work for at least 8 weeks, which means that you can set up your cards at the beginning of July and they will keep going and trapping thrips throughout the entire period of this high thrips pressure. Now there's an ongoing debate over which color of sticky card Western flower thrips prefer. Some studies have found yellow is better and others have found blue is better. Well, a new study that came out this year might have found the reason for these conflicting results. It turns out that different Western flower thrips populations can develop different color preferences. They tested the color preferences of a population of thrips from Germany and a population from the Netherlands. One preferred blue and the other preferred yellow. So that then begs the question, which color do thrips in Niagara prefer? Previous work by Omafra found that yellow cards caught more thrips than blue, regardless of the manufacturer. During the study, these greenhouses had more than 85% Western flower thrips, but the card counts weren't differentiated based on species. In my 2019 survey, which did look at both species separately, onion thrips preferred yellow at all 3 locations that I sampled at, and Western flower thrips preferred yellow at 2 of the 3 locations. We don't exactly know what was happening at the 3rd location. I suspect maybe lighting or greenhouse cover had something to do with it, but we don't really know. But the moral of the story is that around here, yellow is the best choice overall, although, with some variation based on location. But unless you're going to run your own trial in your own greenhouse to confirm which color works best for you, just stick with yellow. Okay, so the long-term goal of my research is to create a more effective biocontrol-based IPM strategy for onion thrips and Western flower thrips. And in order to do that, we need to know what biocontrol tools aren't working. Believe it or not, these pictures came from a grower that was doing all the right things IPM-wise. They were using a tried and true thrips biocontrol program, and it had been working really well for Western flower thrips. But then something changed, and then they started taking heavy damage. At some point, their thrips population had changed from mostly Western flower thrips to a predominantly onion thrips population. There was almost 80% onion thrips in these hard-hit varieties. What they didn't know was what it was about their IPM program that was failing. And they've been using a very typical IPM program, which is often referred to as the standing army approach. Regular releases of predatory mites, weekly biopesticide sprays, and nematode applications are the core components, although there are, of course, other options and combinations, and then each grower might have a slightly different recipe. Because Thrips IPM programs are multifaceted, it's not immediately obvious which component is the weak link here. So to figure out why this program was failing to control onion thrips, we have to examine each component one by one. Now, there are plenty of studies already that show that the bios that we use for Western flower thrips do work for onion thrips. But the thing that's missing is comparative head-to-head trials. So we actually don't have any way of knowing whether they work equally well or not. The experiments I conducted on the biocontrol agents and pesticides in my master's project were done in lab trials. On the face of it, lab trials might seem like a strange place to start since they're fundamentally unrealistic. But the real world is made up of like hundreds of different factors, so it's really hard to pin down exactly what is causing the effect that you're observing. So we need to start in the lab and look at each factor in isolation before jumping to greenhouse experiments or commercial trials. Lab trials also represent the best case scenario. If a product doesn't work in a situation when we can control as many variables as possible, then it's not likely to work in a more complex environment once climate, plant structure, and other organisms start interacting with the thing we're trying to test. And since no one has done these comparative trials before, I needed to start at the beginning of this process. So my first trial looked at predatory mites, which are arguably the star of any Thrips IPM program. For this experiment, I wanted to find out if there was a difference in the predation rate of different mite species on Western flower thrips compared to onion thrips. In this trial, I tested Cucumeris, Swirsky, Degenerans, and Lomonicus. I put a single mite in a container with 8 thrips larvae and saw how many thrips the mite ate in 24 hours. And this was a no-choice trial, so each mite was only given a single thrip species at a time. Generally speaking, the mites I tested ate at least as many onion thrips as they did Western flower thrips. In Swirsky's case, they actually ate more onion thrips than Western flower thrips. When comparing the mites to each other, Swirsky ate 68% more onion thrips than Cucumeris did. So Swirsky might be a better option for onion thrips, but this has to be validated in a greenhouse trial first. I also ran the same kind of trial as a choice trial, in which I gave Cucumeris and Swirsky equal numbers of each thrip species. Typically, onion thrips and Western flower thrips are both found in the greenhouse together. So if mites do prefer Western flower thrips for some reason, then that could explain why they don't perform as well for onion thrips as my previous no-choice trials suggest that they ought to. But what I found was the complete opposite. Both mite species ate more onion thrips compared to Western flower thrips. So what this tells us is that predatory mites' preference are not what's causing biocontrol failures. I ran the same kinds of trials with Aureus insidiosus and found similar results. They ate more onion thrips in the no-choice trials and exhibited no preference between the two in the choice trial. So based on this information, Aureus should also control onion thrips at least as well as Western flower thrips. Now, anecdotally, I have seen Aureus be very effective for onion thrips and would really like to test them out in a greenhouse trial to see if they actually are a better option than mites. Next, I tested the efficacy of Bavaria bassiana. The product I used for this trial was Botanigard, and I tested 3 different rates. The label recommends a range of rates, so I used the high end and the low end, and I also tested a rate that was 10-fold lower than the label rate. And again, I found no difference between the thrip species, which indicates that onion thrips and Western flower thrips are equally susceptible to Botanigard. And the last biocontrol agent that I looked at was entomopathogenic nematodes. Although Steiner Nematode is the most often used species in greenhouses, both Steiner Carpocapsae and Heterorhabditis bacteriophora have been shown to infect thrips. I tested all 3 nematode species in a lab trial that used a commercial peat-based potting mix as the substrate. The nematodes were applied to the substrate after thrips began to pupate, and I assessed how many survived to adulthood after 6 days. There was no difference in mortality rates of the onion thrips and western flower thrips for all nematode species, with one notable exception, and that was Steiner Nematode, which is the species most often used in the greenhouse. It actually performed quite poorly against western flower thrips. This mortality rate is lower than what is often reported for lab trials for Feltier and western flower thrips, and my suspicion is that this was due to the potting mix substrate that I used. A lot of research in the literature uses either they put the nematodes directly onto the thrips pupa, or they use really simple fine-grained substrate like sand. But previous research by my colleague Taro Saito used potting mix, and he also found that Feltier caused very low mortality in western flower thrips. I could go off on a tangent, but I won't because I don't have time. But aside from this little mystery, what the data does tell us, back to our original goal, is that nematodes should work at least as well for onion thrips as they do for western flower thrips. So if all of the bios are supposed to work for onion thrips as well as they do for western flower thrips, so why don't they actually work in practice? Well, the actual mechanism that is driving poor biocontrol outcomes could actually be behavioral, either on the part of the thrips or the biocontrol agents. The laboratory assays are conducted in a confined 2-dimensional space, which is very different from the complex architecture of a plant. And lab trials don't allow for some of the behaviors that might affect thrips survival, such as where they like to be in the plant or their ability to escape predators. We already know that western flower thrips and onion thrips have some differences in behavior. Research from Cornell University found that onion thrips and western flower thrips are more active at different times of the day. I had the opportunity to confirm this in the greenhouses at Vineland. Being active at different times of the day is most likely to affect the efficacy of pesticides that rely on direct contact, such as Botanigard. So if onion thrips are not active in the afternoon, and that is when growers are spraying, they might pick up fewer spores compared to western flower thrips. However, this difference in behavior is not likely to affect the efficacy of systemic or translaminar pesticides. Similarly, the location within the plant matters for predators as well. Research, also from Cornell University, demonstrated that Aphidoletes works better for green peach aphids compared to foxglove aphids. And it's not because they have an actual preference for green peach aphids. What it is that green peach aphids are found higher up in the plant canopy, which is where Aphidoletes spends more of its time. We've also seen that there's a location difference between western flower thrips and onion thrips. Western flower thrips feed very heavily on flowers, as you can see in this picture here, whereas onion thrips do not. If predators are also drawn to flowers, this means that they're going to encounter western flower thrips more often than onion thrips. And finally, predator avoidance might be playing a role. When approached by a predator, many animals will understandably attempt to get away, but they don't all do this at the same speed. Research from the University of Florida observed that although western flower thrips and Florida flower thrips spend equal amounts of time in the flowers, when they are approached by Aureus, Florida flower thrips escapes more quickly than western flower thrips. And then as a result, Aureus ends up catching and eating a lot more western flower thrips. Unfortunately, all of those things take more time to untangle than I could fit into a 2-year master's project. But luckily, we have received funding for a second master's project to continue this work. We're still searching for the next student. So if you or anyone you know is interested in pursuing a master's in this field, they should get in touch with Rose Bowdenhouse as soon as possible. Okay, so since we still don't know why biocontrol isn't working, we're probably going to need to use pesticides for a little while longer. But I'm going to start this section with a disclaimer. You might have heard that onion thrips are more susceptible to pesticides than Western flower thrips. I said it earlier in this talk. But as far as we can tell, that's true in general, but it's important to note that onion thrips, more susceptible doesn't mean that they're not pesticide resistant. Onion thrips have a proven ability to develop resistance to many different active ingredients. And studies show that their level of resistance can be highly variable. Take this 2005 paper, for instance, looking at resistance levels to 3 different pesticides in Ontario onion fields. The 8 sampling locations here that I've highlighted are all in the same municipality, but you can see that the resistance varies dramatically for all 3 products. Diazonone, for example, that I have highlighted with the blue arrows, caused 90% mortality in one population, but only 10% mortality in another. Since we think that most thrips are coming from outside, this means that they might be coming in with pre-existing resistance depending on the pest management practices of the outdoor crops in your area. We also usually find onion thrips and Western flower thrips together in the same crop, so we need to take their resistance into consideration as well. Okay, now that that caveat is over, on to my lab trials. I didn't have time to do a comprehensive pesticide screening with multiple populations and several different rates of each product. So this trial was done using using adults and larvae from the onion thrips colony in my laboratory. These thrips were originally collected from a greenhouse that uses primarily biocontrol and had been kept for many generations with no pesticide exposures. So this is a best case scenario onion thrips population, which means that if a trial doesn't work in these lab trials, it's probably not going to work any better in a commercial operation. I tested 5 commonly used pesticides that we know have worked for thrips in the past. I used the highest recommended label rates for thrips in greenhouses, in greenhouse ornamentals for each of the products. The method that I used is a leaf dip method. So I dipped chrysanthemum leaves in these solutions for about 30 seconds and then let them air dry for about 30 minutes. I then added 10 thrips onto the leaf in a container, and then I counted the number of live and dead thrips after 2 and 5 days. So these are the results at the end of the experiment at 5 days. The x-axis here is percent mortality, so 100% means that all of the thrips died, as was the case with success. The dotted lines show the cutoffs for what is considered control versus suppression versus partial suppression. As you can see, success was the only one that reached the level designated as control for both adults and larvae. Both Pylon and Ference meet the cutoff for suppression, but Belief and Pontos performed pretty poorly. However, in addition to the mortality they caused, Ference and Belief caused feeding cessation, which put an immediate stop to damage, which is beneficial in and of itself. But also, eating is pretty essential to life, so these products could also result in higher mortality over the long run that just wasn't captured in a 5-day trial. Since the overwhelming majority of growers in Canada are using biocontrol, they're likely looking to find a product that is both effective against thrips and doesn't obliterate your biocontrol agents. In terms of efficacy, success came out on top. The caveat here, of course, again, is that this is only tested on my laboratory population, so you might get different results depending on the levels of resistance in your thrips population. And even if you are getting high mortality with success, we don't want to overdo it. When success first hit the market, everyone was really excited about it, but Western flower thrips developed resistance in record time. So if you want to make sure that these products work when you really need them, rotate your pesticides, use biocontrol as much as you can, follow all those resistance management tools that you should all be familiar with. So after success, Ference and Pylon both delivered a decent level of suppression. To figure out compatibility with bios, I did a literature search as well as consulting the BioBest and Cobert side effects databases, and then compiled all of this information to create a very general overall compatibility rating. Overall belief seems to be soft on most biocontrol agents. There is very little information on the compatibility of Ference though. It's believed to be soft, but this is mostly anecdotal information. So if preserving your bios is super important to you, it's probably better to tread carefully and assume that they might be slightly to moderately harmful until we know more. Contos and success are both slightly to moderately harmful depending on the species of biocontrol agent in question. So again, if you have bios that you're particularly concerned about maintaining, consult the side effects databases. And Pylon, however, even though it worked well, is really hard on bios and likely won't suit most IPM programs. So it's probably only useful if your crop is at a stage where you don't care about preserving your bios. And we also want to consider damage cessation. Visual damage is obviously important to ornamental crops, but again, probably will contribute to their long-term mortality. I observed damage cessation in both belief and Ference, but not Contos. And since success and Pylon had a really high mortality rate in my trial, it's difficult to judge whether this took place or not. So when you put this all together, there isn't a product that was the best in all three categories. I would say that success and Ference offer the best balance of efficacy and compatibility, but each grower will have to figure out what suits their own crop best based on the stage of their own crop, based on what other pests they need to manage, how important it is that they don't mess up their biocontrol program, etc. So if you are planning to spray for onion thrips, there are some things that you can do to make sure you're getting the most out of your sprays. Step one is to identify which species are in your crop and figure out what the proportions are. We don't actually have a threshold for what proportion of onion thrips you should have at this, and it's probably going to differ depending on the crop, but anecdotally, once you reach more than like half onion thrips, you probably need to do some kind of intervention to avoid excessive damage. To get a good sense of whether or not the sprays are working, at least working the way they should, do standardized plant taps before and after spraying to make sure that the thrips population is actually decreasing. We have found that sticky cards aren't always reliable for assessing the resident thrips populations, so plant taps are the best way to quantify the thrips population that's actually in the crop. And lastly, whatever pesticide you choose, you probably want to reapply bios after spraying unless it's the end of the crop cycle. Keeping in mind that a product can still cause up to 25% mortality in your biocontrol agents or reduce their fitness in other ways and still be given the not harmful designation. The most important part of this process, in my opinion at least, is the standardized plant taps, because this is pretty much the only way you'll know if your sprays are actually doing what you think they are for sure. To do this, you'll sample a predetermined number of plants. So let's say you tap 10 plants per bench, spaced evenly apart down the bench. And then the actual number of plants themselves isn't super important. What matters is that you sample the exact same number of plants each time. So you'll tap the plant gently but vigorously over a white tray and collect all the thrips that fall out. You want to count all of the thrips that you collect, and ideally you also want to ID which species you have and assess the proportions. For identification purposes, if you have collected a bunch of thrips, a subsample of about 50 to 100 thrips is good enough to give you a good idea. Then you can repeat this at weekly intervals before and after your sprays. Because resistance changes over time, you want to do this every time you decide to spray. Because just because it worked in the past doesn't mean it's going to work again in the future. It might seem time consuming to do this process every time, but spraying is also time consuming. Also, it's expensive. So I think that you probably want to make sure that it's actually doing what you want it to. Okay, so for an example of what this looks like in practice, let's take a look at a case study. This is the same cut mum grower that I referenced earlier that was doing all of the right things in terms of biocontrol and ended up with mostly onion thrips. They ended up deciding to spray Ference, and they let me come in to collect some data before and after those sprays. Of course, we had started by identifying their thrips. They had almost 80% onion thrips in the hard hit varieties, but they actually had a better balance of thrips in some of the other varieties. So they actually only spot sprayed the varieties that had this really high concentration of onion thrips. I did standardized plant taps before and after spraying, and using this data, I was able to determine that after 2 sprays, they had reduced their onion thrips numbers by 50%. Meanwhile, their Western flower thrips numbers were completely unaffected. Compared to the results of the lab trial, in which I had 58% to 81% mortality, these results are actually quite reasonable for a commercial trial. So spraying wasn't able to eliminate their thrips problem, but ultimately the grower was happy with the results because it saved their crop from disaster. They did some more sprays after this and their onion thrips numbers declined further, but they still have an established population. So as suspected, these sprays gave them suppression, but not eradication. And this case study is a really good illustration of our current onion thrip situation. Pesticides can help you avert disaster when onion thrips get out of control, but it's not a sustainable long-term solution, which is why we still need to find a way to improve biocontrol so that we can avoid getting to this point, hopefully, as much as possible. Okay, so let's recap the main takeaways from my research. So this study was the first survey of thrip species we conducted on Ontario greenhouses in 30 years, and we found that onion thrips are prevalent throughout the Niagara region, and we found them in multiple species of ornamental crops. In general, Western flower thrips are still the dominant pest species, and onion thrips make up about 30%. Observationally, higher proportions of onion thrips are usually associated with more severe thrips damage. Sarah and I developed a simplified thrips key that growers and consultants can use to ID their own thrips. Knowing which thrip species you have can help you determine the best IPM strategy, track changes in the thrips populations over time, and being able to ID thrip species also helps growers intercept invasive species early so they can take action sooner rather than later to prevent them from establishing in their greenhouse. When it comes to infestation sources, my data suggests that most Western flower thrips, and definitely onion thrips, are coming from outside. Large populations of Western flower thrips in the landscape indicates that they are now able to overwinter in Niagara, which was not the case 30 years ago. We confirmed that the thrips found on chrysanthemum cuttings were Western flower thrips and didn't find any onion thrips coming in from this source. Peak thrips pressure for both species is in July and August. Western flower thrips pressure declined with the outdoor populations, but onion thrips persisted in long after the outside pressure tapered off. Mass trapping is a really simple tool that you can use to reduce how many of these invading thrips make it to your crop. Our research confirms that both onion thrips and Western flower thrips prefer yellow cards in general over blue cards in this region. In my laboratory trials, predatory mites and Aureus insidiosus all ate as many or more onion thrips compared to Western flower thrips. And this was true whether they were offered one thrip species at a time or given a choice between the two. I also found that onion thrips and Western flower thrips are equally susceptible to Botanigard. Similarly, when comparing the efficacy of 3 different entomopathogenic nematode species, I found that they all worked at least as well for onion thrips as Western flower thrips, if not better. Steiner Nematode, which is most commonly used for thrips, performed poorly against Western flower thrips in my trials, the reasons of which aren't entirely clear. The other species tested, Steinernema carpocapsae and Heterorhabditis bacteriophora, worked much better for Western flower thrips, and they were just as effective for onion thrips as Feltier was. Until we figure out better biocontrol options, pesticides are what we have to work with to suppress major outbreaks. In lab trials, success in Ference seemed to offer the best balance of efficacy and compatibility with biocontrol. In a commercial case study, Ference offered suppression of onion thrips, reducing populations by 50% after 2 sprays. However, the Western flower thrips population in this greenhouse was not affected. Before you decide to spray, you should identify your thrip species and determine the relative proportions. As a general rule, if you have more than 50% onion thrips, you probably want to spray to avoid excessive damage. Although onion thrips appear to be more susceptible for now, onion thrips also have a documented history of resistance to many different pesticides. So the efficacy data here might not reflect the way that your population will respond. The susceptibility of thrips within your greenhouse can also change over time. And so for this reason, you want to do standardized plant taps before and after spraying to make sure that the products are actually working as well as you expect them to. Okay, so back to this flow chart of how we test pest management tools. Biocontrol passed the first phase. Everything I tested performed at least as well against onion thrips as it did for Western flower thrips in the lab. So now we need to scale up to cage trials to see if they continue to work once they're used on a crop. My master's project is complete, but we now have funding for a second master's student who will carry on this next phase, and hopefully we can figure out what is actually the weak link in our biocontrol strategies. This issue of no one paying attention to thrip species composition isn't just a problem in greenhouse ornamentals in Canada. For such an important pest, there's actually shockingly little data of this nature in any crop globally. There's really only a handful of studies around the world that have done these kind of surveys that have been published. So we want to expand out this survey to take a look at other greenhouse crops to see if onion thrips are a problem in greenhouse vegetables, greenhouse strawberries, cannabis. Some recent data from the United Kingdom showed that there are a number of non-Western flower thrip species that affect strawberries. And anecdotally, we have found that onion thrips is the primary thrips species in cannabis in Ontario. So these crops might also be benefiting from further research on greenhouse IPM for onion thrips. Beyond the next master's project, a topic that needs a closer look is vent screening. Since most thrips are coming from outside, vent screening would be the most effective way to reduce thrips pressure. Screening is much more commonplace in warmer climates than it is in Canada. But when pepper weevil arrived in Leamington, veggie growers began screening their vents, and they didn't experience any negative consequences of this, no increase in diseases. But the added bonus is that they noticed that screens reduced other pests, such as aphids. So some of the key research needs, one of which is to identify the barriers that are preventing ornamental growers from installing screens. There's surprisingly little data on the cost-benefit analysis or estimates of the reduction in pest pressure that screening provides. And this is necessary information to determine if the upfront costs are worthwhile for growers. And so since we need to know these things, if we want to figure out what's going on with screening and why growers aren't doing them, I'm going to get Sarah to pop up a little poll for you and yeah, collect some data immediately.
Oh, 1 second while I change the poll type.
Okay.
All right, everyone. Yes, you're going to be our first little guinea pigs. So please answer accordingly. Basically, we're concerned with why people aren't using screening. We don't see a lot of it. Don't be shy.